Good day, and welcome to the final day of PAG 2022 and our final two plenary presentations. So it's my privilege right now to introduce Dr. Elinor Karlsson. Elinor completed her PhD in bioinformatics from Boston University. Following that, she held a postdoctoral fellowship at Harvard University, and in 2014 started her own research group by joining the University of Massachusetts. And Elinor is currently Associate Professor in Bioinformatics and Integrative Biology at the University of Massachusetts. And she's also Director of Vertebrate Genomics at the Broad Institute of MIT and Harvard Medical School. Elinor is a specialist in mammalian comparative genomics, as you'll see today, I'm sure, and participates in a very large international project called Zoonomia, and is also the founder and the chief scientist behind the Darwin's Ark project. And I would encourage people to visit Elinor's website to read a lot more about those fascinating projects, huge data sets which are being generated. Elinor, we look forward to your presentation today around comparative genomics and what's coming in the future.
Thank you very much. Let me just share my screen here. Okay. Okay. Hopefully it's all working now. Somehow I got muted again. Thank you for the wonderful introduction, and I'm sorry I am not able to join you, everybody in person, but at least we can do something virtually. So I'm going to talk about the work we've been doing recently in comparative genomics and kind of what we're thinking about going forward. A lot of this work is going to focus on our project Zoonomia and what we've been finding with that. This was a project in comparative mammalian genomics where we've generated new genomes for 130 species and then aligned those genomes along with everything else we could find. So we got about 240 placental mammals in there and scored sequence conservation. And we published an overview of this data set in late 2020 in this paper that was in Nature. But as you might have noticed from my affiliations at the UMass Chan Medical School and at the Broad Institute of Harvard and MIT, I work at two places that are very concerned mostly with human health. And they think that animals are very nice, but only really mostly as they're concerned in human health. So it's kind of interesting doing comparative genomics, comparative mammalian genomics at a place like that, because you're kind of looking at things from both perspectives. But the great thing about comparative genomics is that there is really a very clear relevance to the question of human medicine and human health. As most people watching this presentation probably already know, our capacity to sequence genomes has obviously been skyrocketing for a while now. The Broad Institute, for example, has this large genomics platform where they have lots of sequencers. And I think as of at least, September of last year, their capacity had reached about 1 human genome every 3 minutes. And so that's a 3.3 gigabase genome. And so we've gotten very, very good at producing a lot of genomic data. But the problem that we have is that we still don't actually really understand what most of it does. And so we kind of get these really big files out, these really big data sets, and we're still trying to figure out how we actually figure out what's doing something. And in a species like human with its 3.2 gigabase genome, one of the biggest problems we have is that for most of it, it may not be doing anything. The human genome is about 1.5% coding sequence. And the rest of it, we know that some of it is obviously functionally important. You've got all the regulatory landscape and all of that. We don't think the whole 98.5% is important. And so even figuring out what's doing something is a really big challenge. And so this is kind of where the human health part of it comes in, because people have been studying human genomics for a while now, and they've been finding things that seem to be associated with disease, but figuring out which positions in the genome, not just regions, but specific positions are the functional positions and actually also might be good therapeutic targets remains an ongoing challenge. And there's a number of very large projects that have been looking at how we actually assign function to the genome. There's the All of Us project, the ENCODE project, NOMAD, GTEx, the UK Biobank, et cetera. And actually, NIH recently recognized this problem with a big new initiative to look at the impact of genomic variation on function, the IGVF consortium. And to this end, we've been sequencing an awful lot of humans. I made this graph about a little over a year ago, and just kind of looked in the sequence read archive and summed up how many sequences, how much genomic data there was from humans in there, and then compared that to how much we have from other species. And basically, we've been doing a really, really good job of sequencing an awful lot of humans and not a particularly good job of sequencing anything else. There are obviously species that are better represented in that, but I was really struck by that comparison that even species you might think have a fair amount of data, we're still looking at an awful lot less than what we have in humans. And in my mind, this is a problem because while some of the differences between humans are obviously important, for the most part, humans are mostly the same. You know, there's many different ways of measuring how different two genomes are. But if you just look at, single base variation, for example, it's, I think, about approximately 1 every 1,000 bases or so. An awful lot of the sequence, when you're sequencing humans over and over and over again, you're sequencing the same thing over and over again. And the question that we're kind of asking ourselves now is that by sequencing just one species and a species that doesn't have a huge amount of diversity over and over and over again, what are we actually missing in the genome? And as I said earlier, this is kind of the human genome. We think that some proportion of that 98% is actually functional. And this was really kind of not first illustrated, but nicely illustrated back in 2011 when Kerstin Lindblad-Toh and her team at the Broad Institute published the 29 Mammals Project, where they sequenced, I think, 29 mammals in addition to humans, lined them all up and basically looked at what wasn't changing as quickly as you might expect over evolutionary time. And what they found was that they were estimated at that point in time that approximately 7% of the genome was conserved. Very interesting. And they also found at that point in time, coming back to that human health question, that if you looked at that 7%, it tended to be much more enriched for variants that contributed to the heritability of human disease than other functional annotations of the genome, suggesting that it was actually a really good mark for something that, if you're actually looking for the functional variants that explain human disease, focusing on these conserved regions that were identified in the 29 Mammals Project would be an effective way of doing that. But there were several notable limitations of the 29 Mammals Project. The first was that with only 29 mammals, they had a false discovery rate of about 10%. And the other was that they couldn't actually tell you exactly which position in the genome was conserved. They could get it down to about 12 base windows, which if what you actually want to do is find functional variants, that can be a hurdle. And so what we kind of asked at that point in time was, could we actually, with a big enough data set, with a well-designed data set, identify exactly which single bases in the human genome were conserved? And for those of you that aren't used to thinking about genomes this way, the basic logic that we're thinking about here is that genomes mutate. We know that, that's just what DNA does. And over the 100 million years since placental mammals diverged, essentially all of the possible mutations in the genome have actually occurred at some point in time, and repeatedly. And so when you actually find a position in the genome where you line up a whole lot of placental mammals and you don't see any variation at that position in the genome in all of those species, that doesn't mean that that position hasn't ever changed over 100 million years. It means that any individual that had a change in that position hasn't left any descendants. And so that's actually a really interesting functional mark. It means that changing that position is a really bad idea. And in order to identify these positions, we basically, as I said, need to line up all these species and identify what's not changing as quickly as you might expect over that 100 million years. And in order to do this, the key metric is branch length. So branch length is a number that basically represents the average number of nucleotide substitutions per genomic position in your alignment. And so in my little toy example here with 8 species, I've labeled this position as having have a branch length of 2. So basically on average 2 nucleotide substitutions per genomic position. The higher you can get the branch length, that means the more random mutations you're expecting to see at any given position. And that means you're going to have a lot more power to discern which ones are unexpectedly not varying. So if you saw, and for example, in my toy example of 8 species, you saw a position didn't have any mutations in it, you're like, oh, it's only 8 species. Maybe it just happened to not happen in these 8. But if you get up to hundreds of species and you're still not seeing any changes, then you start to get really interested. So more genomes, you get longer branch length, and you have more power. And so we did at the beginning of this project, a kind of back of the envelope calculation to basically say, what is our estimated number of false positives given a certain branch length? And so, and this by false positive here, we mean something that is conserved across all the species in the data set by random chance. And so with the 29 mammals project, we had 29 samples. I think it was actually 30 with the human, and that gave us a branch length of 4.9, which was an expected false positive in the genome of about 23 million out of the whole human genome. And with the 200 mammals project, when we got up to 241 samples, it's actually 240 species. So this is actually based on our real data set, which, spoiler alert, has 240 species and 241 samples in it. We had two samples from one species, has a branch length of 16.6. And that actually gets us down to an expected number of false positives of 191 total in the whole human genome. And so, just for a bit of fun, we did a comparison of using the same statistic to the Nomad project, which is this huge project where it's actually bigger now, but at the time we looked at this, they had about 70,000 human exomes. This is just the coding regions of the genomes aligned. And that turned out to give you a branch length of about 0.17. And if you kind of extrapolate out your expected number of false positives in the genome, it's very, very big. And that's just reflecting the fact that, if you get a whole lot of humans and you line them all up, because most humans are mostly the same, you're not expecting a whole lot of variation. And so figuring out which positions have unusually little random variation is very hard. And so we've been now from this kind of back of the envelope calculation, we have the 200 mammals project. And so this was actually the brainchild of Sherston Lindblad-Toh, who's really kind of seen it all the way through from the beginning. And this was a goal basically of resolving sequence conservation to a single base pair. It turns out it's not actually 200 mammals, it's 200 placental mammals. This was a very conscious decision. It's not that we don't like the things that don't have a placenta. There was actually science behind it. And so basically the placental mammals diverged, or they're also called the Eutherian mammals, diverged about 100 million years ago. And so this is actually enough time for sufficient variation to have arisen for us to be able to detect those unusually conserved positions. But it's close enough in terms of evolutionary time that the regulatory elements in the genome are more likely to be conserved. And as I mentioned at the beginning, we can identify pretty well the coding regions of the genome, but it's identifying the functional regulatory elements that are the real challenge. And so we wanted to make sure that we were able to capture those. And so at this point in time, we've got all sorts of caveats on our 200 mammals project name. It's not just 200, it's placental, it's 240, et cetera, et cetera. And so we decided it would probably just be easier to come up with a new name at that point in time. And that is where we came up with the Zoonomia project. So this was actually named after a book by, I think it was Charles Darwin's uncle, Erasmus Darwin, many, many years ago, before Charles Darwin. And it seemed like a very appropriate name for our project. I was recently looking at Charles Darwin's On the Origin of the Species for something else I was doing, and discovered this nice little quote where he basically said that he greatly admired at one point in time, but when he went back and looked at it, he was very disappointed because the proportion of speculation was very large in proportion to the facts. And so I'm hoping that we haven't, somehow cursed our project to have way too much speculation relative to facts based on, naming it Zoonomia. One caveat on this project, and this is obviously something that will get better in the future, is that at the time we were making these genomes, genome technology was not where it is now. It's amazing how fast things have changed, and we needed to make a lot of genomes that are relatively low cost. And so we used an approach, which was an Illumina sequence data, short read data only, assembled with this Discover de Novo software. And it worked beautifully for our purposes. It had the advantages of being very low cost, requiring very little DNA, and not needing high molecular weight DNA. And this was really important because that branch length statistic that I mentioned, number of variants that you expect per site, in order to maximize that, even within the placental mammals, it's critical for us to get as much diversity as possible. We want to maximize that branch length, which means sampling as many of the kind of families of mammals as we possibly can. And it turns out that an awful lot of those are very hard to get samples from. And so by needing very little DNA to make our genomes, we were able to expand not just the number of species in our alignment, but also the diversity of species in our alignment. But of course, as anybody working on genomes now knows, that a short read genome does lack long range contiguity, which has some limitations as far as what we can do with them. But some of them have been upgraded. Harris Lewin has done this really nice study looking at the kind of chromosome evolution. They upgraded them with kind of proximity ligation, and the others, some of the others have actually been upgraded at this point in time as well, because genome stuff changes really fast. Okay, so this is just the point that I just mentioned. We did manage to get a lot of diversity. We added 43 families that didn't have genome representatives when we started the project. And in our current alignment, there's only 22 families of placental mammals that aren't represented at all, which was fantastic. And I just want to give a shout out now to everybody who provided samples for the 130 new genomes that we made, but especially San Diego's Frozen Zoo, which is run by the San Diego Zoo Wildlife Alliance. And basically they've stored, I think it's over 10,000 primate fibroblast cultures for a huge diversity of species over many decades. And that resource proved to be absolutely invaluable as far as trying to get a diverse set of mammals for this project. And I think about half of our 130 new genomes were actually made using samples just from the Frozen Zoo. And I also want to point out that at the end of the day, there were 36 species that we ended up dropping from our list because we couldn't ever actually get a DNA sample from them, even though we required a really low quality and low amount of DNA to make the genome. Okay, so these were the available placental mammals when we started. I should actually put a year on this one. This is our current mammalian tree. So we filled it out an awful lot. It's a really nice data set. We've got, as I said, 240 different species of mammals in there. And I just wanted to just kind of stop briefly and mention these new resources that have been being built by the UCSC genome browser people. As we add more and more species to genomic resources, to GenBank and everything, anybody who actually wants to do anything with these genomes will be very familiar with the problem of actually needing some annotation and some resources to be able to use them. And so that's what they've actually been building at the UCSC genome browser. This was work that I think was led by Karlsson. And basically they've got right now over 1,800 track hubs, and that includes gene and repeat annotations for each of those assemblies, and then also the ability to do searches on them using PCR and BLAT. And most importantly, and I wanted to make sure everybody knew this, if you have an assembly that you want to have in the UCSC GenArc and it's not in there now, you should email them. As long as you have a GCF identifier, they should be able to help you. And so all the information is on here, or I'm sure that you can find it on their poster. Okay, so I've now talked about the design of the project, what our goals were, and all of the beautiful genomes that we made that were short read, but still beautiful. But of course, then we had the challenge of aligning them. And this was, as it turns out, a huge challenge. And so we used a new way of aligning genomes. So it's basically, we use this software called Cactus that was developed by Benedict Patton's lab and run, well, actually developed and run by his graduate student, Joel Armstrong, who since graduated. And the really cool thing about Cactus is it's what we call a reference-free genome alignment software, meaning it doesn't use human as a reference genome. And so this is got a huge advantage. And so basically what Cactus is doing is it's rather than taking human and aligning each of the other species back to human and seeing, basically, do you align to the human genome or not? It's taking each of the species and sort of reconstructing the ancestral genome at each node in the mammalian tree as you go up. And so every species kind of gets equal weight in this final alignment. And this means that we can find things or we can see signals that are just not apparent if you're dealing with a human referenced alignment. The most obvious example is that if you have some kind of important genomic element or functional genomic element that's been lost in the human lineage, then your human referenced alignment will not include that element, and you can't actually study it in any of the other species because it's been lost in humans. It also means that you can't actually, it also means that when you have this kind of non-human referenced alignment, you can find things that are unique to other lineages. So you can find things that are carnivore specific, and so that's why they're not in human. And so this actually gives us a lot more opportunities to kind of explore what's in the alignment. Usually when we work with it these days, we tend to work with a referenced version of the Cactus alignment. And so depending on what questions you're asking, you can reference the alignment on a particular species, but the Cactus alignment itself is reference free. So I put the slide in when we first finished the alignment, which did take us a very long time to do. I will point out that Benedict promises me that the Cactus software has actually gotten much more efficient and doesn't take nearly this much time to run anymore. but it got done. And actually the really fun thing that they did, so they were doing our big mammal alignment and they were doing a big bird alignment at the same time. And because cactus does this thing where it's basically reconstructing the ancestor at each node in the tree as you go up, once they had their mammal alignment and they had their bird alignment, they could actually hook them together at the top by reconstructing the ancestor of birds and mammals. And so that paper that I mentioned at the beginning that was in Nature was actually part of a 3-paper package. It was the bird alignment, the mammal alignment, and then a paper about cactus, which is a little confusing because a cactus is also a plant, but you know, it was kind of fun. I did, when this came out, when Benedict first tweeted about it and we had our bird mammal alignment, I went to find pictures that could actually illustrate this because I like pictures, and found this very cool picture of a dog bird hybrid. I will point out it was easy to find because if you Google images to find dog bird hybrids, it's apparently a thing that people make, which I did not know. I did try Googling bird mammal hybrid as a more generic version, but it turns out that if you Google mammal bird hybrids, you mostly find a lot of pages about monotremes. So there you have it. And so now we've got our alignment. What do we do next? What are we actually doing with this cool new alignment that we have, this reference-free and 240 species, et cetera? So we tested for signals of selection. So basically things that are unusually conserved, so changing slower than you expect, and also for things that are unusually accelerated, so things that are changing faster than you would expect. And basically, so we use a software called PhyloP to do it. And there's basically 3 signals that we can get out of this. One is that the position is neutral. So that means that basically we're seeing about the number of changes we'd expect based on our predictions. And our interpretation would be that this part of the genome probably isn't doing anything. I should rephrase that. That the actual sequence of bases in that position, in that part of the genome isn't relevant to the functioning of the genome. The second signal that I've talked about a fair bit is that the position is conserved. So those are places where changes are much rarer than we would expect. And then we would infer that changing this position harms survival or reproduction. And the third is that the position is accelerated. So these are positions where changes are more common than we'd expect. I'm not going to cover that as much in this talk. It's a complicated signature to interpret. It's a little bit prone to being, you know, if you have weird errors in your alignment and things might affect an acceleration score. But we are finding some interesting things in the alignment by looking at the accelerated regions in humans, for example. And of course, one of the things that we did with the neutrally evolving sequence that I'll just mention briefly before going on to the conserved sequence is that we made a tree. And so that is actually a really cool use in my mind of the neutral sequence, because not being a big person on phylogenetics myself, I've long been aware of this problem of making a tree based on anything like a gene sequence where you're expecting there to be evolution acting on it, that it may not be a tree that reflects the kind of true relationship of the species. As far as I can tell, you never have a tree that actually reflects the true relationship of the species. But the idea of making it using neutrally evolving sequence, it means you have a lot of sequence and you can actually specifically define which sequence you're going to use based on its PhyloP score. And so using that tree and PhyloP, I think actually the tree got used by the tree got made after PhyloP. We've identified overall, just based on the distribution of scores, that we think that approximately 13.1% of the human genome is under evolutionary constraint or is conserved more than you would expect by random chance. And so that's based on the overall patterns. As you go across the genome, as you can imagine, if you imagine going across the human genome in terms of the alignment, the depth of the alignment does vary as you go across the genome. There are places that just don't align to other species, you know, places in the human genome that just don't have alignments to anything else. And so that does actually get captured in the PhyloP score, but I wanted to mention it as a slight weirdness of the data set. And so this graph is just showing on the y-axis the percentage of the genome that's covered given the number of species aligned. So, you know, there are no actual zero data species, but, you know, 100% of the genome is covered by at least one species, and then very little of the genome is covered by all species, you know, is aligned in all 241 species. You can see there's this kind of weird little humpy thing going on. That's basically reflects the fact that your alignment reflects the phylogenetic tree. So this is the human line for that graph. And what you can see is that basically almost everything aligns if you stay within the hominids. And then if you go out to the simians, and it drops down a bit, primates, et cetera. And so that's why you kind of have that humpiness to the line. And as I said, so approximately 13.1% of the genome seems to be conserved. We can't that's not the proportion that we can say single bases are conserved. And so I've made this little chart here just to show that within that overall 13.1%, on a single base level, we can score each position at a kind of false discovery rate number. And overall, we defined a constrained base as any position that had a false discovery rate less than 5%. And that gave us about 3.5% of the human genome being in significantly constrained single bases in our alignment. As I said, this is a reference-free alignment. We haven't looked at all 240 species in there, but we have referenced the assembly onto other species, including chimp, mouse, dog, and little brown bat. There's some variation in the amount of sequence that we're detecting as being conserved in each. I don't know how much of that's an actual biological thing and how much of it reflects the species that are in our alignment. And some variation in the overall percent conserved, but it doesn't vary dramatically. I put both the megabases conserved and percent conserved into this because each of these species has a different sized genome, and so the percent conserved doesn't necessarily, equate. It depends which statistic you're worried about. I think little brown bat has a much smaller genome than humans do, for example. Okay. So what do we get out of having 240 mammals as compared to one of the earlier alignments which had 100 vertebrates in it? So we get more stuff that's conserved at a false discovery rate of less than 5%. We get more coding exons that are conserved at that threshold, about 1.5 times more as far as sequence goes. But you can see that our most dramatic increases are within the regulatory type of elements of the UTR exons and things like that. And that was really exciting to us because, as I mentioned at the beginning, that was something that we were really designing this project for. And we also saw, as we kind of hoped to see, that the Zoonomia annotation of conservation is actually extremely enriched in complex trait heritability. So this is just showing a number of different types of annotations across the human genome. The left one is showing the overall enrichment of human complex disease heritability in those annotations, and then the right one is actually saying how much extra information is added by that particular annotation relative, once you've conditioned on everything else that's in there. And what you can see is we're actually doing really well with the constrained in mammals. We're doing even better with the constrained in primates. That's a bit of a complicated data set. So the problem with the primate data set is that we only have 43 primates in our alignment. And while it is a lot of primates, a lot more than we even had mammals in the original 29 mammals project, it's not enough to get anything close to single base resolution. We would need an awful lot more than that. And I think we looked once and it was like, basically, if we kind of got as many primates as we could possibly get, we would kind of have the resolution that we have with the 29 mammals project. So a lot more, but not single base. So that said, overall, the stuff that's conserved in primates is extremely relevant to the heritability for human disease, as you might expect, just them being so closely related to us and having so much of the regulatory landscape conserved across primates. Okay. Oh, and then the other thing we saw was that if you look at both the mammal phylo-P and the primate FAST-CON, so what this is looking at is the disease enrichment in heritability in that annotation versus how much of the genome is conserved, the decile of the genome conserved. And so the decile of the genome conserved is basically the more to the left you are, the more conserved you are, and the more to the right you are, the less conserved you are. And what this is showing is that not only is the conserved stuff enriched for heritability, but the more conserved you are, the more enriched for heritability you are, which is exactly what you would expect to see if this was a useful functional mark. And then the other question, of course, is we found conserved stuff. It seems to be really useful. Did we actually achieve single base resolution? So we've looked at this in a number of different ways to try and figure this out. This picture is just a picture made by my graduate student, Shirley, looking at loss of function variants. So these are coding variants in the human genome, and just, what does constraint look like around those positions? And you can see it does really nicely peak in that loss of function position, and we're getting much higher phylo-P scores than we did with the earlier data sets. But we can also look at coding regions and at the codons. So codons have a known degeneracy pattern where the first position, there's usually only one option, but by the time you get to the third position, many of the amino acids have many different nucleotides that will work equally, mostly equally well in that position. And so we actually do see, if we look at the constraint in those codons, we can pick out the three that have aren't completely degenerate at that third position. So the tryptophan and the methionine, you can see, actually have a very different pattern, as well as the stop codon, suggesting that we are getting that resolution at a single base sort of level. We did do that across all of the variants. It showed that there was a significant correlation there between degeneracy and your phylo-P constraint, or sorry, conservation. And we also did kind of find some fun patterns looking at, say, methionines, which can be a start codon, but may also not be a start codon. And if you are a start codon shown here in red, you tend to be more constrained than if you're not actually a start codon methionine. And the same thing with cysteines. If you're active in a disulfide bond, then you're going to be more conserved than if you're not. And the same thing with the heritability enrichment. I really love this plot made by Stephen Gazal. And basically what it's showing, if you look at a constrained base, so this is one of these ones at an FDR of less than 5%, and then you kind of look around at the heritability enrichment really clearly peaks at that center constrained base position and then drops off as you move away from it. And you can also see the primates here, and it's mirroring what I mentioned earlier, which is that they tend to have higher heritability enrichment in the primate annotations, but we don't have the single base resolution in the primates, which is why you see that plateau across the top of the green line there. And as you know, just as kind of an additional add-on to this, I'll mention that we did identify a new set of ultra-conserved elements that we called ZooUCs. So the original ultra-conserved elements were 481 regions greater than 200 bases that were identical between human, mouse, and rat. And these were identified back in 2004 by Gil Bejarano and his colleagues. And so the ZooUCs we defined, because we have so many more species in our alignment, we now defined as regions that are greater than or equal to 20 base pairs, where every single position in those 20 or more bases is identical in at least 95% of the species, which is 235 out of 240. Did I get the 95 or is 98%? Just go with 235 out of 240. And that gives us about 4,500 ultra-conserved elements. And out of those, I think about 700 of them overlap with those previous 481, and then about 3,800 of them are new. And then finally, we did try to go back and see whether we could define whether the conserved positions were actually functional using some really nice data that got published using massively parallel reporter assays to look at the 3' UTR of about 20, I think it was 20 disease-associated regulatory elements. And so, well, first we just made a really nice picture because I like pictures. This is the 29 mammals data for the promoter of LDLR, that gene. Each of those lines is a single position. That's our 100 vertebrates data. You can see that the constraint is the conservation scores are going up. And then this is what we get with 240 mammals. So we've got a much stronger signal of conservation at the conserved positions there. But the really fun thing is when you compare it to those MPRA results. So the massively parallel reporter assays is basically an assay where you take an oligo containing your variant of interest, and then you have an oligo that has your variant of interest mutated. You put both of those upstream of a reporter gene and screen and basically look at whether that reporter gene gets expressed or not. But because the reporter gene is sequence barcoded, you can do this, in parallel and test tens of thousands of oligos, kind of in a single pool. And it basically what it's telling you is in this very artificial kind of assay, changing that position does have an effect on gene expression. So it's basically kind of an assay for function. And so the red lines here, basically, the more it goes down, the more of an effect that variant is having on the expression of the reporter gene. And the one on the top, the blue, the phylo-P, the higher it is, the more conserved that position is. And you can see a really nice, at least in this one example, they're almost mirror images of each other. And then we zoomed in on this one region here and actually looked at ClinVar variants. So these are variants in humans that have been connected to some kind of disease. And so what we saw was that there were 3 variants in that region that had at least 2 lines of evidence suggesting that they were pathogenic. And those are those 3 red boxes in there. And you can see each of those positions is actually both highly conserved and seems to have an effect in the Kircher et al. MPRA assay. The final position that has that really tall Zoonomia phylo-P score was actually not in ClinVar. It's not in anything because basically there hasn't been a variant reported in that position in humans ever. So, you might hypothesize that that's just a position that we don't see varying because it's very functionally important, or we just haven't seen it yet. you will be unsurprised to know that I cherry picked out of that paper the one that was the prettiest picture for the one that I just showed you. That was the LDLR one. I put a red box around it. We do see the same correlation at many of the other UTRs that they tested in that paper. And I've just given kind of a set of at least 12 of them here. And we did do an overall test of all of that to basically look at whether we saw a significant difference here between the actual position that had an effect on the expression of the nearby gene and the things to either side of it. And what you can see is that especially at the more conserved positions, the one that is a much higher proportion of the things that are actually having an effect on gene expression tend to be very highly conserved as compared to the things on either side. Okay, so that was a fun, little take on what's been going on in the conservation world. But I just wanted to step back a little bit and talk about non-human species. And so in the Zoonomia paper, we did ask this question about what value a single reference genome has for biodiversity conservation and management. This is work we're trying to build on now to make it a little bit more robust, because we did find a really interesting pattern. The nice thing with the Zoonomia data is that we had these 130 genomes that we'd made using the same basic genome assembly approach. So this is all of our data, and I'm basically comparing how much homozygosity there is in the genome on the y-axis, and then how much heterozygosity there is in the genome on the x-axis. We're not doing regions of homozygosity because we have short contiguity genomes, so instead we had a metric we called segments of homozygosity, which is sort of similar. And basically, what you're expected on this plot, we did see that they tend upper left-hand corner. And this really suggested that there might be information even in a single genome. So you're never going to be able to classify a population as being at risk based on a single genome. And I don't want to imply that, but we're getting better and better and better at sequencing genomes now. And what this is suggesting is that you might be able to sequence the genome from a population, an individual or several individuals from a population, which compared to a lot of the other things you have to do to survey populations is pretty straightforward right now, and at least potentially prioritize which populations you need to be extending a huge amount of resources to actually figure out what's going on there. And in the last few minutes of this presentation, I just wanted to kind of mention briefly this project we did early in the COVID outbreak, which I think is a really nice example of the potential for genomics to inform, things going on in the world around us. So when the coronavirus pandemic started, it became clear very early on that other species were susceptible to getting infected by this virus. And we were approached by many people in the kind of animal care world, zoo and conservation communities, who really wanted to know which of their species they needed to protect the most. And this was a really hard challenge, because while there are many systems for assessing whether a species is susceptible to being infected by a virus, most of them are incredibly labor intensive or very difficult to do with natural populations, et cetera. And how do you actually assess hundreds and hundreds of species for susceptibility to a novel pathogen quickly enough that that information can be useful to the people that are trying to protect them? And at the same time, there was interest from the human research side as well in kind of model systems. And we were sitting, obviously stuck at home, as many of us were back in March and April of 2020, which seems like a long time ago now. And we were like, the genome, we have a huge amount of genome data for lots of species. There's so much data in GenBank now for sequencing many species. Can we do anything with it? And can we actually make a prediction as to which species are likely to be susceptible? And I just want to kind of step back. This is our collaboration here. It was a fantastic thing to be doing at that point in the pandemic, because, it was a very weird time. And this project was really kind of initiated by Harris Lewin and led by Joanna Demis, who brought it to a kind of paper in a remarkably short period of time. It wasn't a complicated analysis that we did. I think many people in this community who do things with genomics will recognize that. We basically took the spike protein, so the thing sitting on the surface of SARS-CoV-2 that binds to ACE2 that actually infect our cells. And we looked at the binding domain between the host ACE2 and the spike protein. It's 25 amino acids, about 75 bases of DNA. And we weren't actually asking which species was the best fit between those two. What we were asking is, in which species does ACE2, the binding domain of ACE2, look the most like humans? Because we knew that humans were really, really susceptible. So the hypothesis was, the more you look like a human, the more you're likely to be susceptible. And we also did some kind of protein structural modeling at the same time to kind of confirm what we were finding. In total, we had 410 vertebrate species in there. I think we've gotten it up to 500 and something now, just basically by going in and scoring new species as we found them. The first thing we saw was that everything that was very high, or medium, we basically classified it into 5 different risk levels. We only saw mammals in those first 3 groups. The very high group was all the catarrhine primates. There was nothing else in there. And these guys were very high risk because basically they all look exactly like humans. And this included the gorilla that we knew that we'd been seeing infections in already. So sorry. The risk species was a much more interesting and much more diverse group. And interestingly, this included the white-tailed deer, which we now know seem to be very susceptible to being infected by this virus, although they don't actually seem to be getting particularly sick or at all. They seem to be mostly asymptomatic. And then we had a group of species that were very low risk. And this included Mus musculus and its closest relatives, which, at least with the original strain of SARS-CoV-2, were not susceptible to infection. So it was nice that we got that right. But it also interestingly included the mink, which we got, based on this, very wrong, because the mink were definitely susceptible to infection, and we had these huge outbreaks going on in mink farms. We still don't know exactly what happened here. It's an interesting situation because while pet ferrets, who are related to mink, do seem to be susceptible to infection, they don't seem to be super susceptible to infection. And so this may be some combination of they're more susceptible than we predicted, but also the actual environment in which those mink are living maybe have a big influence on whether they're getting sick or not. It does vary by clade. So we can see that the bats tend to be very low risk. We did see some evidence of more positively selected variants in that binding domain in bats, which kind of an interesting signals. The carnivores are mostly low risk. That yellow wedge in the carnivores is actually, I think, mostly the big cats. And those we've had a number of different reports and illness in big cats, as well as less so in small cats. Okay, so that was a very cool paper. We got it out. It was actually very rewarding because one of the organizations that I work closely with is the Franklin Park Zoo, and they and other communities were able to actually take this information on board and try to use it to help protect their animals. As I said, it was not a perfect metric, but it was a metric. And given that it's so hard to measure infectivity, it was some information. But I do want to point out that this is not a finished problem. And while we now recognize that the deer are at risk and surveillance is starting in deer, we still don't really understand which species are actually susceptible in the real world. And there doesn't actually seem to be very much surveillance going on of a broad spectrum of mammals, which is what we should be doing if we actually want to know what's getting infected with this virus. As the deer showed, you may not actually know where it is until you really start looking. Okay, so I'm not going to talk about the work that we've done on forward genomics, but I do want to call it out because it is a really interesting part of this project. Moving away from conservation, the question is basically, can we study cool things that other species can do that humans can't? And one of the classic kind of fun examples of this is hibernation, where species that hibernate manage to kind of get super obese, become very, very inactive for a number of months, lose all that weight, come out again in the spring, and seem to be almost entirely unaffected by this. And many parts of that process would actually be potentially very damaging to human health. And so understanding how hibernating species manage to hibernate would be really cool. And so forward genomics is basically an approach where you say, you kind of look at how species are evolving over time, and then at the end of it, what you're looking for is some kind of signal. So in this case, it's a lack of variation in a set of species that's scattered across the tree, but happens to share a phenotype. And so if you're saying that if they're mostly unrelated, but we see that they look more like each other, that this particular gene or this particular regulatory element than you would expect by random chance, given that phylogeny, this might be an indication that they actually share some phenotype. And as I said, it's something we're working on. And the reason I'm mentioning it here is just because I think it's one of those examples in genomics where genomics is not the hard part. And so the genomics, we've pretty much got that covered. We're going to have way more genomes. We're going to have lots of new genomes. It's going to be super fancy. We're going to have lots of data. It's great. The methods are a challenge. They're not where they need to be yet, but it's something that we're thinking about, and many other people are thinking about how you actually do forward genomics better, both on coding and on regulatory elements. But I think the huge challenge, honestly, is going to be the phenotypes. I mean, it's hard enough to phenotype and individuals within a single species, but how do you actually make sure that you're defining even a phenotype like, you know, one of the ones that we were looking at was the number of turbinals in your nose, which is something you can at least count, and that we managed to do. But, you know, even a single phenotype, how do you actually make sure that what you're measuring in each species is actually comparable when you're talking about hundreds and hundreds of different mammals? And I think it's one of these places where we're going to have to start having fields that are often quite separated from each other really start talking a lot more. And then, of course, just to end this, and I'm sure everybody, given it's a plant and animal genome meeting, will probably be on the same page with me as this, is that even though we are getting much better at sequencing species and the number of genomes that we have from other species is getting bigger and bigger, we're really being dwarfed right now by human genome sequencing. And it's going to be interesting to see in the next few years as we kind of push to get the amount of data we have up, what we're going to be able to do with it. And actually what we can learn when, rather than focusing on humans, we start looking at every species as a potential kind of model species. So thank you very much for listening to me. And I just wanted to give a big thanks to the huge consortium people that's been working on the Zoonomia project, and especially Sherstin Lindblad-Toh, who got the whole thing started. And then the people who are the leads on the papers, several of the big papers that we've been working on: Matthew Christmas, Stephen Gazal, Irene Kaplow, Jen Meadows, and Pat Sullivan, and everybody else all over the world that's been working on this with us. So thank you.
Darren, I can't start my video. Elinor, I assume you can hear me?
Yes, I can hear you.
So yes, thank you very much for that fascinating, very, very huge and rich data set. I'm sure it'll take years to get through it. So we do have a couple of questions in the Q&A that I'll just ask them out here. So early on, there was a question, are there transposable elements limited within certain mammals? And the reason for the question is that many of these functions, many of these transposons, of course, act as enhancers, which is the hardest element to identify.
Yes, so David Ray really led the work on looking at transposable elements in all of our genomes, and he's got some really interesting results showing that different transposable elements are active in different lineages. You've got some particular ones that are more active in some lineages than others. I think bats maybe have a lot more transposable elements than the other lineages, and so we're definitely seeing exactly what you would expect as far as an awful lot of diversity in the activity of the transposable elements. We can also see, for example, that if you're looking at, I think it was regulatory elements, I'm trying to remember which kind. Anyway, I think it was the enhancers, but basically in primates, for example, all of the new stuff is coming from transposable elements. And so if you've got a new enhancer in a primate, it's coming from, or in the primate lineage, it's coming from a transposable element. So they are going to be a huge driver of recent evolution in various lineages, including the primates and especially regulatory evolution.
Great, thanks very much. So there's a question about the cactus alignments. I was also intrigued by this. So are there any cactus alignments for plants made, or are you aware of anything in the plant world?
I'm not. You think you would, given that cactus is a plant. I generally stay away from plants because those genomes are just way too complicated. No, but I think it's actually a really interesting challenge. I know that they've been working on doing a new alignment of a kind of high contiguity vertebrate genomes, and I think it would be really similar to look at doing something similar in plants. I don't know enough about the diversity of plants to know how feasible it even is, but I don't see why it wouldn't be. As I said, you just need to be able to reconstruct those nodes. So if you had the species and the genomes and they were high enough quality, you should be able to do it. But as far as I know, I don't think anyone's doing it yet.
Good, thanks. Just watching the time, Elinor, I'll just go with one more question. When you construct phylogenetic trees, what kind of SNPs did you use? Synonymous or non-synonymous, genic or intergenic? Did you get the same
so
tree? Did you
yeah,
get
so
same
for the
tree?
phylogenetic tree that I showed, we were using only, so we didn't actually worry as much about whether it was genic or non-genic. I should actually check whether they specifically excluded genic stuff, but what they were only including things that were neutrally evolving. So we actually used the phylop scores themselves to define what parts of the genome we would use to make the phylogenetic tree. And so I can't remember the exact amount of sequence that went into it, but it was quite substantial because most of the genome is neutrally evolving.
Right, okay. So we're a little over time. I think I'm going to have to cut it off there, folks. I want to thank Elinor, fantastic talk, and we look forward to as you dig deeper into this this data set and through it. And
Thank
I'll be
you.
moving. Thank you very much, Elinor.
Thanks.